Recombinant DNA is DNA in which the nucleotide sequences from two different sources are combined with the same DNA molecule, and this can result from a specific type of genetic engineering, and it's called molecular cloning. We'll discuss that in a little bit, and just like a relatively common alternative is polymerase chain reactions, which will be covered later by DNA cloning is, DNA, or wait, the hereditary information that is needed through the DNA is called a gene, and the genes are very hard to find in the DNA. The specific genes needed in DNA cloning normally constitute for about 1/100,000th of it. Like, this is just an example, but it's normally something with a really small ratio like this, and it's also, or because of this, it will be incredibly inefficient to clone the whole chromosome, and in addition to this, it is also very difficult once it is copied to discriminate between the genes and the rest of the DNA. So because of this, scientists have developed a more efficient way of doing this, and it is called gene cloning. Cloned genes are useful to make copies of specific gene that a scientist would want that would be useful, or to make a sort of protein product. They can be used in basic research. One example would be to see if something is resistant to pests or something like that. Or they can be transferred into another species. Would anyone want to guess how a product of that, or how that can be done? No, okay. If a human were to, this is kind of a stretch, but if a human were to want to have wings, they could theoretically put a wings gene in humans. I've been stretching that a bit, but it's an example. And then they can be used sort of medically with how I mentioned with a sort of protein product. Like, they could mass produce a specific type of protein product, like human growth hormone or something like that. This is a diagram of sort of how they would do something like that. While it's up here, does anyone have any questions thus far? Yes, Sam?
How come it doesn't work like that though? Say if we wanted wings, how come we can't just put?
Well, it's there's a lot. I'm not exactly sure if they are able to yet, but there'd be so much you'd have to do so much of this in order for that to happen, because there's going to be many traits for wings. There's going to be a lot of genes that would be needed to have. So
I feel like there would also be a lot of mutations, so that they wouldn't even bother to get for example wings. Be perfect wings that go out and actually achieve flight. It'd be near impossible, the odds of it actually growing to the right size and the right shape.
In order to do this, there's some steps you would have to take. First, you would have to remove bacteria and their genes from the plasmids, and normally they do this from E. coli, because their plasmids have they're set up so that it is sort of ideal for this. They carry two genes that are sort of necessary for this whole process to happen. Both plasmids and the DNA are then digested by an enzyme that cuts the DNA at multiple sites, creating fragmented DNA, which makes it able to be put back together. We would then mix the human DNA fragments with the cut plasmids, and this allows for base pairing between their sticky ends. Then we would add the DNA ligase, which permanently joins the two together. And after this is done, the prepared DNA is mixed with bacteria, and this makes the gene unable to hydrolyze lactose, and this makes it unable to mix with any more DNA. So there's like no more like additional genes that could screw it up. Steven or Jim?
So the Human Genome Project is a mapping project of the human genome begun in 1990s. This effort was completed in 2003 when the nucleotide sequence of the vast majority of DNA in each human chromosome are discovered. And the way of like research the DNA, this is one of the way for research the DNA. So it's called Saucer burning of DNA fragments. This application is to detect specific nucleotide sequences within a DNA sample. In particular, this is useful for comparing restriction fragments. A restriction fragment is a DNA fragment that is resulting of the cutting of DNA strand by the restriction enzyme. So it produces different sample of genome DNA. So the method is DNA or RNA sample is placed on gel right here, and the restriction fragment in each sample are separated by electrophoresis, forming a characteristic of general band like this. Then, if you play like when the things are like placed like this, if you press the weight from upward, the alkaline solution will go upward and will copy on the nitrocellulose filter. Then the separate DNA will appear on the here like this. And when you're using the probe, you can find specific DNA fragments that you want research about. So this is the way that you find the DNA or RNA fragments. So if you get RNA like this, you can separate this on agarose gel like this. Then it will transfer to the membrane, which is like when you place apparatus like this. Then it will reveal on here like this. And if you put probes, that will show on the film like this. So you can find a specific RNA they want to discover for research. And this is the other way to research the DNA or RNA. And this application is used the sequence to find the sequence of nucleotide in any clone DNA fragment. So there's a lot of fragments, and it can determine like any fragment by the special machine that can reveal the products of the length of DNA or RNA. So the fragment of DNA is like incubated in this cube like this. So this is the original DNA, and this is DNA polymerase, and this one is the primer. So the synthesis of each new strand starts at the 3' end of the primer and continues until the nucleotide is inserted at random. So set of labeled strands of various lengths will generate like this. So this is 3' end, and this is the prime end. This primer is based to stock with destroyed primates, and these are all like colored one, so that you can detect after you copy the other DNA like fragment like this. So the labeled strand, like this one, the other DNA, is just like this. In the mixture of like separated by passage through a polyramide gel in a capillary tube like this. And this movement through this machine will reveal on the print like this, so you can identify a nucleotide and its end by the spectrum.
I'm covering the practical application of DNA technology, and many fields benefits from DNA technology and genetic engineering. For example, medical application in pharmaceutical products and forensic evidence and genetic profile, and environmental cleanup and agricultural application. For medical application, one benefit of DNA technology is identification of human genes in which mutation plays a role in genetic disease. Scientists can diagnose many human genetic disorder by using PCR and primers corresponding to clone disease genes, then sequencing the amplified product to look for the disease-causing mutation. And for pharmaceutical products, advances in DNA technology and genetic research are important to the development of new drugs to treat diseases. Pharmaceutical products that are protein can be synthesized on a large scale. For example, in protein production in cell culture, whole cell culture can be engineered to secrete a protein as it is made. This is useful for production for insulin, human growth hormones, and vaccines. And protein production by farm animals and plants. Transgenic animals are made by introducing genes from one species into the genome of another animal. Transgenic animals are pharmaceutical factories, producers of large amount of otherwise rare substance for medical use. Farm plants are also being developed to make human proteins for medical use. This is an example of farm animal. They extract medicine from goat's milk to make medicine, and it became legal like in 2009, so they do a lot of that. And in forensic evidence and genetic profiles, like you can use an individual's unique DNA sequence to identify the person. Genetic profiles can be used to provide evidence in criminal and paternity cases and to identify human remains. Even more sensitive is to use the genetic markers called short tandem repeats, STRs, which are variations in the a number of repeats of specific DNA sequences. PCR and gel electrophoresis are used to amplify and then identify STR of different length. The probability that two people who are not identical twins have the same STR markers is exponentially small.